Good afternoon, everyone. My name is Dave Arnold, the Executive Director of NextGen Precision Health Initiative. It is a pleasure to welcome everybody to the next of the Discovery Series. This is our flagship seminar every month. This month we have a very far-traveling speaker from Finland. So it's a great pleasure to welcome Dr. Johanna Laakkonen. So Dr. Laakkonen is a vascular biologist interested in vascular anomalies, cell-to-cell crosstalk, and leveraging that crosstalk to design viral-based gene therapies to hopefully improve these conditions. So Dr. Laakkonen is an Academy Research Fellow at the University of Eastern Finland's AI Virtanen Institute of Molecular Sciences. Dr. Laakkonen did her master's and PhD in molecular biology at the University of Jyväskylä in Finland, and then did a postdoc fellowship in adenovirus vectors. So I'm very familiar with adeno-associated virus vectors, but they're actually using adenoviruses, and also studied the blood-brain barrier. We had the opportunity to walk through the building and show them some of the facilities in this building, which I'm very proud of, that we have all the way from clinical research down to cell-based therapies where we can have it all in one place. And really, Dr. Laakkonen's research is starting in the clinic and working back to basic mechanisms and then hopefully going back to the clinic. So this really epitomizes what we're trying to do with precision interventions for big health problems. So it's a great pleasure to welcome you, and thank you for coming all this way. I understand that you arrived on Sunday and have had some opportunity to explore around to nearby cities, so welcome to Columbia.
So hi everybody, it is my great pleasure to be here today. Thank you for a kind invitation and an introduction. So my name is Johanna Laakkonen, and today I will be describing you how paracrine factors should be taken into account and targeted in next generation therapy for vascular anomalies. So my objective during the next hour is to give you first an overview of vascular anomalies, and then I'm going to share you our exciting data how we think fibroblasts induce venous lesion formation. And lastly, we are going to discuss about therapies, especially gene therapy, and how it could be beneficial for treatment of many vascular diseases. So vascular anomalies occur due to abnormal vascular development. So it can affect any vessel type and locate anywhere in the body. Vascular anomalies are classified based on their histology, biological behavior, and clinical presentation to vascular tumors, which grows by cellular hypoplasia, and vascular malformations, which represent more a localized defect in vascular morphogenesis. So today we are going to focus on two types of vascular anomalies. First of all, sporadic venous malformation, and then provisionally unclassified vascular anomaly termed ASD, angiomatosis of soft tissue. So sporadic venous malformation is the most common type of vascular anomaly. It contains enlarged venous vessels. It's a low-flow lesion, and if found near skin, it is of bluish color. Usually when these venous malformations are found, they are already bigger than 5 centimeters in size, and typically they locate in the head and neck area, but can locate anywhere in the body. So when VM is pressed down, it will often shrink like a balloon losing air. And then it fills back up again when you stop, like a balloon filling air. So this is the result of blood being pushed out of the malformation and then refilling slowly. So that's why as a first-line treatment, often compression garments are used. Symptoms of these venous malformations include pain, swelling, disfigurement, and functional impairment, depending on the location. And PIK3CA and TEC somatic mutations are found in these lesions in endothelial cells. Usually, venous malformations are managed throughout life, so goal is more to alleviate the symptoms. So symptomatic lesions are treated with sclerotherapy, cryotherapy, endovascular laser treatment, or surgical resection. And then also sirolimus is used as off-label. And this is also now tested in a phase 3 clinical trial. So in sclerotherapy, needles are used to deliver a medicine called sclerosant directly into the abnormal veins of the venous malformation. And this then damages and destroys the abnormal vessels. The goal of this sclerotherapy is to cause the malformation to scar that little or no blood will flow through the venous malformation. And this will then cause the VMS to shrink. However, several sclerotherapy treatments are often needed, and they are usually at least 6 weeks apart. Thus, sclerotherapy makes the VM get smaller, but they can get bigger again over time. So then angiomatosis of soft tissue are provisionally unclassified vascular anomalies, and they typically locate in lower extremities or trunk. ASD is not found in skin, and the lesions are subcutaneous or intramuscular and can cross tissue planes, and this makes their management also more difficult. Mesenchymal tissue components are often present in ASD, especially muscle infiltrating fat. MRI findings overlap with that of venous malformation. Here are images of malformations in the lower extremities. And however, the treatment is usually surgical resection, as sclerotherapy has shown to be an inefficient treatment. So that's why differential diagnosis is important. Symptoms are the same, so pain and functional impairment, as with the venous malformation. So histologically, ASD is defined as a different entity than VM. So whereas venous malformation consists solely of venous structures, in ASD there are vessels of different origin and size. So there are dilated venous channels in which the smooth muscle cell layer ranges from thick to very thin, and there are lymphatic structures as shown here in brown, and artery-like vessels also shown here in these confocal microscopy images, where the smooth muscle cells are labeled in red and endothelium in green. And here you really can see the smooth muscle cell hypoplasia and also these very narrowed lumens. There are also clusters of small arterioles, venules, and capillaries, which are surrounded by intramuscular fat and fibrosis. What is interesting, vessel morphology also changes according its tissue microenvironment. So here are images from the same patient that had intramuscular ASD lesion. And here in the first, you see the vessels lining nicely up in the resection margin. So this is normal skeletal muscle vasculature. Then when you go in the area that there is abundant fat, you see that the vessel hierarchy changes. It's very disorganized. And in the intramuscular lesion area, it's more organized, but there are definitely pathological angiogenesis in that area as well. So what about then the somatic mutation in ASD? And this was not known until we recently studied this in our patient cohort. So the demographics of our patient cohort are shown in here. So in this study, we had 20 ASD patients. These were mostly intramuscular lesions and located in lower extremities. And the patient had pain, swelling, and functional impairment as symptoms. So we did first whole exome sequencing and then validated the data by droplet digital PCR. And we found out that majority of the patients had PIK3CA missense mutations. The most common one was in the location of H1047R, which we are now focused in this presentation more. And this is found in the kinase domain of the PIK3CA gene. The mutation was only found in endothelial cells and not in stromal cells. So what is this PIK3CA? It is a plasma membrane-associated kinase, and its signaling is activated by growth factors, cytokines, or hormones. The signaling cascade activates AKT mTOR genes, which results in cell growth and proliferation. PI3K has 3 subunits, P85, P55, and P110 catalytic subunit, which is the one that our mutation locates in. So as both intramuscular VM and ASD can have the same genetic mutation in PI3K, why VM is responsive to sclerotherapy and ASD is not. And we got kind of the same idea as Eero Saarinen had, a Finnish architect that designed the arch in St. Louis, as he had stated that every object, large or small, has a relationship to its neighbors. If it's a building, the townscape will affect the solution. So we thought maybe lesion growth is associated with, besides the mutated cells, to genotypically normal neighboring cells and tissue microenvironment. And it has been known actually for a while already that mutated cancer cells secrete proteins that attract tumor-associated cells, such as macrophages, endothelial cells, and fibroblasts. And these tumor-associated cells then in turn secrete proteins that stimulate cancer cell proliferation, tumor angiogenesis, and recruitment of cells to the tumor site. Cancer-associated fibroblasts are predominant components of the tumor microenvironment, and they are known to promote endothelial cell activation and proliferation, for example, by secreting vascular endothelial growth factor, VEGF. Cancer-associated fibroblast CAVs have been studied now with single-cell RNA sequencing, which brings kind of order into the chaos of the cellular diversity by clustering cells to their subtypes by detecting their MRI expression levels. Cancer-associated fibroblasts have been suggested to originate mostly from the normal fibroblast, but also from the tumor endothelial cells. So these cells are highly heterogeneous, and generally, CAVs have been suggested to create a tumor-permissive microenvironment, activate the normal fibroblast, and promote angiogenesis by crosstalk with endothelial cells, so the neighboring cells. They also affect the immune cells, and now they are studied as a therapeutic target or prognostic factor in cancer. So fibroblasts in vascular anomalies are known to be present in VM, so this is after sclerotherapy, and in ASD prior treatment. Traditionally, fibroblasts are defined as connective tissue cells. They are matrix-producing or remodeling cells that are involved in formation, maintenance, and degradation of the extracellular matrix, ECM, and thus contribute to the mechanical properties of tissues. They are also known to contribute to angiogenesis through modulation of the ECM network that is necessary for formation of the blood vessel lumen. And after injury, they are known to secrete a wide range of mediators. So how does this cell communication work with secreted factors? So in paracrine signaling, molecules only affect target cells in close proximity to secreting cells. And examples of paracrine signals are, for example, synaptic signals of neurons, nitric oxide signal in blood vessels, or blood clotting. And here you can see some of the cellular processes paracrine mediators are involved, such as survival, contractility, angiogenesis, differentiation, or remodeling. And some of the paracrine mediators listed, so there are a lot of growth factors, including fibroblast growth factor, VEGF, and IGF, but also then matrix metalloproteinases. So what about then paracrine effects in vascular anomalies? What is known? They have not been studied yet that much, but VEGFA is known to be upregulated in infantile hemangioma, which is a vascular tumor. And it is actually expressed in stromal cell mostly and hemangioma-derived stem cells. Increased VEGFA plasma levels are also observed in these patients. VEGFA is also upregulated in rapidly growing or reoccurring arteriovenous malformation. And in there, they have been shown to be upregulated in endothelial cells, subendothelial layer, and brain parenchyma. So based on this, our hypothesis was that maybe, similar as in cancer, crosstalk of endothelial cells and fibroblasts could have an effect on PI3K-driven vascular lesion formation via paracrine factors. And targeting of these factors could be then used as a novel therapy for treatment of vascular anomalies that has this fibrous component. So to understand first the endothelial-driven mechanisms, we isolated endothelial cells from patient lesions and did CD31 selection. Following bulk RNA sequencing of patient cells showed that the genes were associated with biological processes shown in here, such as ECM organization, angiogenesis, and cell proliferation. What got us really excited were the signaling pathways, as in here it was shown that EGFR pathway was, and its components were upregulated, and especially TGFA, which is known to be a pro-angiogenic factor and associated with tumor angiogenesis. We further observed this in patients that had only PIK3CA mutation in this H1047R location, and it was upregulated in these patient cells as well. We further validated this result in vitro and by using HUVEC cells or primary endothelial cells and transduced these cells with lentivirus vectors expressing PIK3CA gene, either wild type or the mutant form. And TGFA was further shown in these upregulated as well. You can also see here in these images that PIK3CA causes, so here's the mutated form, wild type and non-treated control. So it causes cell size increase in these mutated cells. In the bulk RNA sequencing of these, we could further show that PIK3 signaling, endothelial cell proliferation, migration, and ECM organization were affected as expected. So finally, we showed expression of TGFA in both ASD and VM samples, and it was expressed in endothelium, but also in these intravascular areas. Also, EGFR was activated, as shown in here in red, thus showing that this pathway is active. So to study the role of TGFA more, we did further siRNA experiments using a co-culture of fibroblast and endothelial cells. So we did so that we first transduced the endothelial cells with lentivirus vectors and then did endothelium-specific siRNA with the TGFA. And this showed us that indeed the cell proliferation in these mutated cells was reduced and due to the TGFA inhibition. So to study in detail stromal cells then in patients, we isolated the CD31 negative cells and characterized them a little bit further, showing here in this Western blot data that they were CD31 negative, alpha SMA positive, vimentin positive, and we are now further doing single-cell RNA sequencing of these patient cells to show the fibroblast subtype. But what we did here, we used this fibrin bead assay model, which has been established in Hughes lab, in which we put HUVEC cells on top of these collagen-coated beads, then embed them in a fibrin, and then put fibroblast on top. And these were non-stimulated cells, so we used primary fibroblasts or then the stromal cells derived from the patients. And we were really excited to see that this was enough, so there are no somatic mutations. So it was enough to have the stromal cells from the patient lesions to induce endothelial cell sprouting. And this got us thinking more about the paracrine factors and what could then induce this effect in endothelial cells. And sure enough, when we then did ELISA from this media, we could see that both TGFA and VEGFA protein levels were increased. We further showed that there are this synergistic effect between the factors. So when we stimulated the cells, primary endothelial cells, with TGFA, we could actually get more VEGFA secretion. And same again with the fibrin bead assay. So when we did treatment of the cells with either growth factor alone or then together, so then we had these both growth factors. There was more increased sprouting as shown in here. So to further understand whether fibroblasts could induce now lesion growth in mice, we first set up this mouse xenograft model to study the venous lesion growth. So in here, again, we used our lentivirus vectors, transduced the cells, mixed them with matrigel, and then put to the nude mice and followed the lesion growth. And as you can show here, when we had the mutated form of the PI3K, the lesions grow. And with the wild type, there's almost no lesion growth detected. Same shown also in here, so wild type on the left, and then mutated cells showing the lesion formation on the right. And we are now also doing ultrasound to more precisely measure the lesion volume. Also, to enable faster analysis of the histological samples, we set up a new image analysis tool called Pro-Angio, and this was actually just published. So it's an open source, easy to use, step-by-step guideline we have included in the manuscript. So it's easy to do image analysis without any programming background. So with this, you can, for example, detect vessel volume, sprout amount, number, lumen size, cell nuclei amount, intensity, and area. And we used as an example image here, so this is the plug, HE staining, showing the red blood cells, and here you can see how it nicely automatically detects the erythrocytes in these lesions. So this makes our life much easier as the imaging analysis is now faster. So you may want to try it out. So then next, we of course wanted to test the fibroblast in this setup. So we used the same kind of outline of the experiment. So we first transduced endothelial cells with the PI3K, but then put in the mix the fibroblast. And this was put to the nude mice, and we followed the lesion growth. And as you can see already here in these images, so this is the same image as before, and here with the fibroblast then, that already with the wild type, there was some lesion formation, but then with the mutant form of the PI3K, there was much more vascularity. Same can be seen here in these histological images. So the fibroblasts together with endothelial are shown here on the right, and you can see already from this HE staining that with the mutant form of PI3K, there were much more erythrocytes present. Also, there were more CD31 positive structures, which was also interesting, shown here in this lower panel, that when there were PI3K mutated endothelial cells present and fibroblasts, so the lumen size started to increase. So this went kind of well in hand with the in vitro results that people have shown before, that fibroblasts are actually needed for the lumen formation. So our working model at the moment is that we think first mutated endothelial cells secrete TGFA, which is then bound to the receptor in the neighboring stromal cells, which then further secretes more TGFA and VEGFA, and this goes further back to the endothelial cells and induce the endothelial cell proliferation and angiogenesis. So what is then the clinical relevance? Can these ErbB inhibitors or EGFR inhibitor, which is the receptor of TGFA, so can this treat a pre-existing lesion? So this we studied first in vitro and showed that the ErbB inhibitor afatinib reduces VEGFA secretion in cells, and also, of course, that the EGFR activation was blocked. We then moved again to this fibrin bead assay model. So we stimulated the cells with TGFA and VEGFA, and then put afatinib there, and we could see that it was reducing the endothelial cell sprouting almost totally. As a control, we also checked that EGFR is expressed in these mouse lesions. So here you can see that when there are mutated PI3K with the fibroblasts, it is highly expressed. So then we moved to in vivo experiment and used the same mouse model as before and let the lesions grow. So we had fibroblasts and endothelial cells which had mutated PI3K. We let these lesions grow for 9 days to see that the lesions are formed, and then we treated the mice with afatinib for 9 days. And we could see nicely that the lesion volume was decreased. Also, vascularity was decreased, as shown in here, also in AG staining, and here in the lower panel with CD31 staining showing in red. So this proved us that ErbB signaling indeed mediated vascular growth in the presence of fibroblasts. So multiple strategies for targeting the PI3K pathway are under investigation, as this pathway is activated in many diseases. So it's not only the venous malformation, but it's also found, for example, in lymphatic malformations, and then HHT, and many other specific venous malformation types. And these have different mutations, so there are different pathways that can lead to hyperactivation of PI3K pathway. So various pharmaceutical approaches have been tested. 3 of them are shown in here. The first is alpelisib, which targets the PI3K kinase directly and this B110 subunit. Then we have miransertib, which then targets AKT. And the most used is sirolimus, which targets the mTOR. However, it is already known that if treatment of sirolimus is, for example, discontinued for some reason, the lesion reoccurs. So it shrinks the lesion, but it doesn't cure this. There's a clinical phase 3 study ongoing, the sirolimus treatment of venous malformations. Also, safety can be of concern, especially for pediatric age patients when long-term treatment is required, as this drug is taken daily orally. So how to then achieve a long-term inhibition of blood vessel growth? And we believe the answer is gene therapy. So gene therapy is a technique that uses a genes to treat, prevent, or cure a disease. So you can introduce a new or modified gene to help treat a disease. You can replace a disease-causing gene with a healthy copy of the gene, or inactivate a disease-causing gene. And there have been already 3,000 clinical trials that have been approved, conducted, or initiated in most fields of clinical medicine. Gene therapy vectors are either viral vectors, which are kind of our favorite, or then non-viral vectors. So these recent successes have laid the foundation for next generation gene therapy. And while these approved therapies have been life-changing to the patients with these specific rare diseases, they actually have offered much broader impact for the field of gene medicine. As an example, there are 2 in vivo approved therapies, Luxturna and Zolgensma. And these use both AAV vectors targeting either retina or central nervous system. And this has facilitated the usage of AAV vectors for other diseases as well. Next generation gene editing based nucleus-based systems to cut DNA specifically are also now developed, but there are a lot of need to still study very fundamental things to develop this gene medicine further. So as an example, function of all the genes are still not known. So we have a lot to do in that area. We also need more knowledge on epigenetic regulators. So these include DNA methylation, histone modification, and non-coding RNA action, as they have been associated with most of the common disease. So these epigenetic changes are reversible and do not change your DNA sequence, but they can change how your body reacts to the DNA sequence. And most of all, we think that understanding how these components function separately is not enough, but we need to understand the tissue microenvironment in each disease. So pro-angiogenic gene therapy have been developed for treatment of ischemic heart diseases, so in which this regional myocardial blood flow is reduced due to the obstruction of coronary arteries. None of the current therapies can totally restore cardiac function, and human heart has a very limited regeneration capacity. So novel treatments are needed for treatment of ischemic heart diseases. Professor Seppo Yläherttola, whom I've been privileged to work with, has been a pioneer in this field, especially by using virus vectors to deliver vascular endothelial growth factors to the ischemic heart. And particularly, he has shown that VEGF-D increases perfusion of the vessels after intramyocardial delivery. So there are a lot of other factors that VEGF studied for gene therapy for myocardial ischemia, and you can see some of them in here, including a lot of different growth factors like VEGF, HEF, and different isoforms of VEGFs. So there have been a lot of studies going on. So when planning a new gene therapy, one needs to take into account many steps. So this starts with questions such as which vector to choose, first of all, where and how to administer vector, but there can also be further challenges like poor gene transfer efficiency or therapeutic response. And this can occur due to the low vector dosage, but also due to the limited transgene expression at the injection site, short half-life of the transgene, or limited secretion of the transgene to neighboring cells, or they can bind to ECM that limits then transgene efficiency. So these can be improved by targeted delivery, or we can optimize administration method and improve vector design, as Nihei Lahankaran was yesterday telling us. Also, understanding of the cell-cell crosstalk is crucial, and understanding the transgene-mediated events and pathophysiology of the disease we want to treat. And we also think that gene therapy in combination with conventional treatments should be considered, actually in many cases. So besides our studies in vascular anomalies, paracrine factors have also been known to have a crucial role in myocardial ischemia and to promote cardiac regeneration in all of its 3 phases, so including inflammatory phase, proliferation phase, and stabilization regeneration phase. And in all of these different phases, there are different paracrine factors that play a role. First in the inflammation, then in the angiogenesis, so in the proliferation phase, and the cardiomyocyte proliferation, and then in the stabilization with the formation of the mature scar. So some of the factors are listed in here. It has been suggested that paracrine factors affect differently to the neighboring cells and distant cells due to the concentration gradient, and thus regulate the tissue microenvironment, but this is not very well yet known. Also, these factors are very pleiotropic in nature, so they induce multiple mechanisms in different types of cells. They can also act as a temporal manner. So what about the next generation therapy for vascular anomalies? So we think that all histological features, genotype, and previous therapies need to be taken into account when evaluating the suitable therapy. So it will be very likely that in the future, for the same vascular malformation, multiple treatment options are needed based on the genotype and tissue composition. So besides drugs that have been developed to inhibit PI3K pathway, we are now developing a gene therapy for treatment of venous lesions that have a fibrous component. So we are doing this by inhibiting paracrine factors, and the aim is, of course, the growth regress, reduce vascularity, alleviate the symptoms of the patients, but most of all, to have a prolonged treatment with a single dose. So we believe that it's not enough to target a single cell type, but target the paracrine factors affecting these cells. And we are currently testing different vectors. We are doing intralesional injections of these vectors and checking the biosafety profile of the vectors to choose the best one. So there's a lot to do, and I'm happy to discuss about this project later on if you are interested. So all in all, we believe that whether you have a good or bad neighbor matters. So identification or characterization of the lesion-promoting cell population provide opportunities to develop novel diagnostic and therapeutic approaches. And we believe that targeting of both intravascular stromal cells and endothelial cells could be a potential treatment strategy for vascular lesions that have fibrous component. And that next generation gene therapy could actually enable a prolonged treatment strategy for various vascular diseases, including vascular anomalies. So with that, I want to acknowledge my group, especially Suvi Jauhiainen, a postdoc in my group, and PhD students Henna Ilmonen and Heta Rasinkangas. I want to acknowledge also the whole vascular anomaly unit and patients in the Helsinki University Hospital in Finland, and especially Päivi Salminen and Pia Vuola. Also, I want to thank Professor Seppo Ylähertuala and Nihei Lahakaram, who has been involved in all of these vector designs and our future plans with gene therapy, and Minna Kaikkonen-Määttä group for sequencing, and of course all the funders who have made this research possible. So thank you for your attention. I'm happy to take any questions.
Okay, great.
That was a great talk. Thank you very much. When you are using targeted gene therapy, do you have positive controls to verify that the agent that you're putting in is getting where it wants?
Yes.
And
yes.
so, for example, when you're looking at venous malformations, do you inject the construct into the systemic circulation, or what are you doing to target it? I'm worried that it's just
Yes.
blowing right by and not getting
Yeah.
where you want it to go. That's the basis of my question.
Yes, exactly. So we are doing intralesional injections directly. My opinion is that local delivery is quite often the easiest and the best. Of course, we are now checking the biosafety profile of the vector, so we see like when we inject into the lesion whether it ends up to liver, spleen anyway, so that's why we are checking now different vectors or whether we have a need to target it. But then we have really nice solutions like what Nihe told us yesterday, that we can specifically target the transgene expression to a specific cell type like endothelial cells if we want to. But now we are going more to this very crude setup just to check which vector is the best at this point.
And then you had pointed to anti-angiogenic therapy in the context of cancer, or I'm sorry, angiogenic therapy in the context of the heart.
Yeah.
But have you considered anti-angiogenic therapy in the context of cancer? Gene delivery to suppress VEGF or to reduce any vascular response?
I think it has been studied quite a lot that people have used like soluble VEGFR2, for example, in cancer therapies, and those are in the clinical trials as well. So it has been studied a lot that if you inhibit the receptor, for example, in cancer, so I think like 30% of all gene therapy clinical trials are involved in that field, that they target the receptor.
And then my last question is, when you're looking at the release of paracrine factors, for example, from fibroblasts, you pointed to VEGF, but they release a lot of other things, as you know, periostin, we can go down the list, okay?
Yes.
Have you used an approach where you're sequestering the release of the putative paracrine factor to suppress the action? Rather than deliver something, sequester what you think may be mediating the response?
Yeah, that's a great question. Yeah, we have now set up a lot of these different co-culture models to see more the paracrine factors and study them, how they are functioning. We've also analyzed in detail the secreted factors that come from the patient cells. I didn't show the data yet, but you're right, there are multiple factors that are definitely secreted, so it's probably more complex story, as always. Yes. But it's very interesting in the future to see.
Thank you. It was a beautiful talk, Johanna. I have two basic questions. The first one, when you show that slide about the clinical trials, and at the end was epigenomic editing. Epigenetics is a field that I'm kind of learning, and it's very fascinated because you can modify, as you said, the expression but not the gene sequence, but is fascinated as is in a specific, because there are millions of genes that they suffer this epigenetic modification. So how are you going to do that, and what are the targets for this editing? So that's my first question.
Yeah, so what we are doing now at the moment, so we are doing the single cell RNA sequencing of first of these fibroblast subtypes to see what they are, and we believe that epigenetics plays a role. So there's a reason why these fibroblasts, remain changed and can then induce endothelial cell sprouting. So we are more focused on that now. So we don't know yet what actually happens. It's very likely to be involved.
Okay.
But we are studying that.
And my second question is sorry, I took a picture of your presentation here about the gene therapy for venous lesions. That graph that you have in one of your latest slides, it shows, if I understood correctly, shows kind of like a feedback loop, correct?
Yes.
What controls that feedback loop?
You mean in the...
Because it's, if I understood correctly, will be between the two types of cells generating a factor, and then going to that cell and then coming back. So what would be controlling that loop and with your therapy? Well,
we think, the mutated endothelial cells, first of all, start to secrete these factors. And the thing is, what is actually quite interesting, what I didn't tell you, is that in these lesions, the fraction of the mutated endothelial cells is actually really small. So there are not much mutated cells. Of course, it depends on each lesion, but let's say 20% of cells are mutated. There are a lot of normal endothelial cells, a lot of fibroblasts and smooth muscle cells, which are all affected. So I think it's quite complex then. We are doing now spatial transcriptomics to see what's really going on with cells, what happens in the more distant cells, what happens close to the mutated cells, to find out how these loops are going. But in principle, we think that that these mutated endothelial cells start to then change the phenotype of the genotypically normal cells. And then they start to secrete more factors that then go back to the genotypically normal endothelial cells and promote actually their proliferation and activation. And that's kind of the thing that then induces the vascularity and kind of boosts the growth of the venous lesion.
You mentioned that the fibroblast induces lesion in vasculature. Mm-hmm. From other hand, there are some documents right now showing that the FGF23, for example, stabilizes glycocalyx, which is very important in vascular health. How we can explain it?
That's an excellent question. I have to say, I don't know. It's an excellent question. We haven't studied glycocalyx. I've been now talking here with different people who are studying it, that it would be super interesting to go more in that direction as well. I think there are many fibroblast subtypes. So it must be related to that. And in each disease, there are many fibroblast subtypes. And they each have their specific function. And nowadays, it's still, fibroblasts do not have very good markers. So that's why we are now doing the single-cell RNA sequencing to see more how these are actually subtyped in our case, in our disease. But I think they will be quite different depending on the tissue type or then the disease you are studying.
Thanks for the great talk. I just have another question, a little more about the genetics. You touched upon it in the beginning of your talk. So the mutations you've identified in these endothelial cell genes, so the affected patients you've been studying, so are they typically homozygous for this mutation? I'm just curious if you looked at other family members who might carry the same defect, are they affected? So penetrance issues. I'm just curious what you see sort of from a clinical genetics perspective in these patients, very interesting patients you're studying.
Yeah, thank you, great question. So we do not study inherited venous malformations. So there are definitely families that have this, but we are mostly focused now to the sporadic venous malformations. So these are much more common, so that's why we focused on those. And in these, so it's only a hotspot mutation in a gene. Yeah.
Any other questions? Yep.
I have a few questions. So, say this gene therapy is successful and it's implemented in the clinic. Mm-hmm. How do you see that fitting in the landscape of current treatments?
That's a great question. So what we are doing actually now, as I was pointing out, that gene therapy could be combined with the conventional therapies. So as kind of now in the field, sirolimus is one of the top candidates. So we are definitely testing our vectors together with that. And also continuing with the afatinib to see like double drug combination. I think it now has been kind of discussed in the field that it may be actually that the patient need more than one drug. So it would be more like introducing more than one and not using a single one. But at the moment, there are no pharmaceutical therapies at all. So these are all in clinical phase so far. Yeah.
So what are the current outcomes? How do they track this as like look at efficacy in a study? What do they just quantify the size or?
Yeah, they quantify the size, but I'm not a clinical clinician. So, but I know that then they kind of also ask for the patients about the symptoms, like whether they see an improvement, and the lesion size is another measurement, but usually it's done by MRI or ultrasound.
Next question, unless somebody else has a question. So these somatic mutations, have you compared sporadic malformations that have the mutation versus not? Do they act differently? Is there something in the genetic milieu of that patient that drives how that mutation shows itself?
Yeah, that's a super interesting question. We are definitely doing it at the moment, actually.
Okay.
So we are comparing in the patient tissue, doing the spatial transcriptomics there.
So you get, going to Dr. Fay's question, do you get the genotype of the patient like like to see if there's any inherited genotype that like drives or changes how this somatic mutation shows itself?
That would be great if it would be tested for the patient. Usually it's not, so we do everything ourselves. So that kind of takes time that we first need to check the genotype. But we focus only to the sporadic venous malformations at the moment. So.
And then I can ask one more question, then I'll hand off. So I've been very involved in gene therapy, mostly adeno-associated virus delivery. So that's a non-integrating viral vector, as we were talking about a little bit earlier. So for this kind of approach, you probably would want an integrating vector, I would assume. But is that your strategy, or are you trying different strategies?
We are trying different ones because it's of course dependent. Like the ultimate goal would be that we would, cure the patient. So we don't know whether it would be enough, like with even using with adenovirus vectors for, that have a transduction efficiency for a week, that if it would be enough together with a drug to reduce it enough. So we are testing different approaches. But yeah, the lentivirus vectors that would integrate that's why it's kind of our top candidates at the moment. So we'll see.
Great, thank you.
Yep.
I have a question. There are congenital venous and arterial hemangiomas that are regressing spontaneously. I'm sorry, I missed the beginning of the conference, but is that correlating with your study in some way, kind of bringing a connection to the therapies and understanding the process of arterial?
You meant whether it's more aggressive type of lesion, and then it relates
the process of spontaneous regression of the congenital hemangiomas that you see a child with a face that it's half covered and then without treatment it's regressing.
Yes. Yes, that's typical for vascular tumors, some of them, like infantile hemangioma. So most of them are not treated, actually, because it's, as you said, you don't need a treatment, and then the patient is kind of cured over time, that it reduces in size. But that doesn't happen in vascular malformations. So these grow in time, all the time, and usually when the patient is a teenager, due to the hormones, or it is kind of thought that due to the hormones, or then it kind of boosts the growth of these vascular malformations even more. So there are different kinds of, but with vascular tumors and the infantile hemangioma, definitely, so they can be reduced, yeah.
So I think that is all we have. I'll turn it over to Dr. Arnold.
So I would really like to thank you for your wonderful talk and thank for everybody's questions. I do have an exciting announcement about next month. So the NextGen Precision Health Initiative Discovery Series is a series that crosses the whole system. So in June 5th, we're actually going to have a live stream here. So please join us for food. But we're also going to have the presentation live in St. Louis at UMSL. So Dr. Rachel Winograd is going to present. So please join us for next month. And thank you for your attention.